All right, so I'm Albert Liu. I'm a graduate student at UC Davis, though, as has been alluded to, I'm from the lab of Patrick Shih, who is now at UC Berkeley. So I'm kind of in this limbo between two of the UCs at the moment. But I'll be presenting on some of the work that I've been doing, a little niche of our lab, and we're investigating Rubisco. So specifically, the work that I'll be showing today will be mostly focused on what I've been doing over this last year and eating up all the shifts at SIBYLS, investigating the evolution of Form II Rubisco oligomeric state. And so a quick introduction slide. This is a lot of graphical stuff, but just blowing through it. Rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase is the full name. It's a primary carbon fixation enzyme in nature. And so we're interested in that in the context of plant synthetic biology and the implications therein from its evolution. And so shown on the left is a figure from a paper in 2008 where they identified the main forms of Rubisco. So this is kind of the classical understanding of the enzyme. And so there are 4 forms that were found in nature previously. This form 1 is the most important and most of interest to people. It's the main photosynthetic form. I have a second structure from form 1 here because this one doesn't do a great job of illustrating the fact that Rubisco, or form 1 Rubisco, adopts this kind of central octomeric core. So it's really just the tetramer of these dimers that are shown in these 2 segments here. And these are capped with these small subunits that sit in between each of those dimers. And then we have form 2 here, which will be the focus of the later bits of the talk. This is typically found in bacteria and is usually in a non-photosynthetic context, so the exact function of these remains unknown. Form 3, in terms of function, the same, but these are typically found in archaea. And then form 4 do not actually perform the carbon fixation reaction, and so they're known as Rubisco-like proteins, but just share the same structural composition. And so this is a slide that Mikhail had shown previously that we kind of glossed over, and I'll do so again. But some of the earlier work from our lab with SIBYLS, we identified this metagenomic form 1 prime enzyme is what we ended up calling it. And so this adopted an octomeric core without the small subunits capping each of those junctions. And so this had previously been unknown and was thought to be an unstable structure. But one of the more notable things and why we prefer to use SEC-SAXS-MALS for these kinds of analysis is the fact that when we were doing native page experiments in the lab shown here in this subpanel D, we saw this compaction upon incubation with a transition state analog. And so initially we thought that this was some kind of oligomeric state shift where potentially more subunits were being added into the cyclic structure and it was increasing in size. But we later found that this was actually due to the expansion of these C-terminal tails that typically enclose the active site when catalyzing the reaction. And so their extension resulted in this increase in size that we observed on native page, but had thought to be addition of completely new subunits. So this kind of keyed us in on the sensitivity of SEC-SAXS for oligomeric state assaying in addition to crystallography. And so we decided to pursue this in the context of different oligomeric states of Rubisco. So this is the entirety of the phylogenetic tree. So form 1s, the photosynthetic ones, are up here. And then this is the clade that we had discovered, form 1 prime, in addition to the remainder. And so for this project in particular, we focused on these form 2 enzymes because early last year, this paper was published in which a group in Israel had characterized kinetically a larger diversity of these form 2 Rubisco that previously were unknown. But one of the limitations of this study, as we found out, was that their assignment of oligomeric state was simply due to or done with size exclusion chromatography. And so that didn't really inform us on anything that may be of interest from this data set. And so we were gifted this plasmid library that was used within this paper. And so what we ended up doing was purifying and characterizing 28 of the extant Rubisco shown around the perimeter of this radial tree or the circular tree. And so this is how you can effectively occupy 20 plus shifts at SIBYLS. But what we discovered was that one of the limiting factors in the Rubisco oligomeric state studies was that previously only 3 crystal structures had been identified from this form. They're indicated with the species names at each of the tips. But one of the considerations that had previously been made was that rubrum here, our rubrum, was the first enzyme to have its structure, or the first form 2 Rubisco to have its structure characterized. And so people had considered this state to be primarily dimeric, and this was an assumption that had carried through to a lot of literature concerning the function of Rubisco. But from our characterization here, most evidently hexamers tend to be the predominant form. They're fairly spread out across this entire tree. All of these samples were previously confirmed to be catalytically active, so they weren't just dead enzymes, but we see that 23 out of our 28 actually adopt this hexameric state.
We have one little interesting thing here that I'll get to in a little bit, a tetrameric enzyme, which also had previously not been known. And then shown on the right are the SAXS curves for representative candidates from each of these states. These were modeled by Daniel, so he's rather the expert in this processing compared to me. But we see here that you do see this kind of difference in trends across the different oligomeric states, which allows for a more sensitive characterization of the different states rather than just using native page or size exclusion. And so moving on to discuss a little bit about the tetramer. So previously, going back to this slide, we really had only identified dimeric enzymes here, hexameric, so a trimer of dimers, octameric, a tetramer of dimers, and so on. But we hadn't really characterized the tetramer yet, and it was thought to be impossible because of this existence of a central solvent channel in all of the cyclic Rubiscos. But what we discovered in combining what we discovered from SAXS with crystallography is that we could in fact have a tetrameric enzyme. Here's a general spinning movie here, but you see that it adopts this kind of twisted interface that isn't quite the same as the cyclic interfaces that we see in different forms. And so this is something that we're interested in right now, and we're doing some follow-up work to better characterize this, and that'll be some of the work that we'll be doing with SIBYLS moving forward. But aside from that, one of the things that we really have been thinking about in terms of the data that we've collected is this kind of evolutionary trajectory of Form II Rubisco and how these assemblies evolved over time. And so one more thing that we did here in all of our experiments was to characterize different ancestral sequence reconstruction enzymes at these corresponding nodes shown on the tree. And so we see this kind of downstream trajectory where the most ancestral form here was dimeric, as illustrated by this gray cartoon up top. And over time, we see that one branch ended up forming this tetramer clade here. We're still looking into this since that is a rather unusual assembly relative to all the other Rubisco that are known. So that'll be something for our future consideration. But from here, we see that there is this kind of downstream evolution where it branches once more. And you see the right-hand side of this entire tree is entirely hexameric according to our sampling. And so it seems that the hexamer was the predominant form here. But we are interested in the left branch here because you see that there is this interconversion event where this maintains a dimeric ancestral state up until a hexameric state. And from there, these will be characterizing in the future as well, just to kind of fill out this tree more. But we do see that you actually do get loss of the hexamerization. And so we're interested in looking at this in the context of whether or not the oligomeric trajectory of Rubisco has been really locked in evolutionarily, as some people have suggested. So there seems to be some kind of a plasticity here, so to say, that allows for these interconversion events based on what we're seeing in the ancestral states. So that was a pretty quick run through of what we've been up to over the past year. Future directions for these things, we are interested in understanding the residues that govern these specific oligomeric states and attempting to control those, as well as I had mentioned previously, filling out this tree more with more biochemical data for the ancestral residue or ancestral sequences. And then some additional tetramer follow-up work trying to understand how that residue, how the interface, has assembled over time, and then mutating specific residues in there to see if we can either break the tetramer into stable dimers or engineer that construct potentially into different dimers. And so I'd like to thank all the people listed here. So I suppose we are a Berkeley lab now. This work was started with a former postdoc, Doug Banda, who's now at AGC Biologics. And I'd like to especially thank Daniel and Mikhail for helping us throughout this entire time. Daniel has been very, very helpful and very receptive of all of our unreasonable requests over time, and Mikhail as well. So I'd like to thank the SIBYLS Beamline for their continued help. And that's pretty much it, and funding sources here as well.
Thank you, Albert. I just have a short question before
Yes.
we move on. So in case you have the identification of different oligomeric state, you mentioned you want to do some mutational study to distinguish which residues are really the interface required for the interface of the specific tetramer, especially that it's something new. And that is how it's going to be important in the carbon sequestration. Do you think you're going to continue that with the carbon sequestration, like kind of assays, or you want to do that SAXS with the carbon sequestration? I mean, in the biological aspects, what do you want to do? This is just simple oligomerization state determination with SAXS, but can you imagine like some kind of bioassay with the SAXS?
Yeah, so I think one of the things that we're hoping to work on more in the future is that, a lot of work has been done trying to characterize the active site of Rubisco and mutational studies therein to affect catalytic activity. But over the past 10 or so years, that's been largely fruitless. And so
Mhm.
we're interested in the idea that potentially something about oligomerization and interface residues can affect catalytic activity. So if we can demonstrate these mutations, then we would also follow up with kinetic activity assays to see what kind of changes are being made at that point. The challenge being that for something like this tetramer, we don't know the biological relevance of this enzyme in the host organism, but the idea that if we can start to identify residues that control the kinetics of Rubisco that are outside of the active site, that could influence future studies for engineering the photosynthetic Rubisco forms.
Great. You can use definitely such just identifying how disruptions happen very easily.
Yeah. So there's something about like distal sites that actually do affect active site performance or control the different reactions that occur in Rubisco.